So welcome to the Environmental Research Seminar. And as always, I am Sung-Kyun Park, faculty member at Epidemiology. And this seminar series is sponsored by the Michigan Life Stage Environmental Exposures and Disease Center, also known as M-LEEaD. So today we have a young speaker, Dr. Anna Young from Harvard University. So Dr. Young is a research associate in the Department of Environmental Health and associate director of the Healthy Buildings program at Harvard T.H. School of Public Health. She earned her PhD and MS in environmental health from the Harvard Chan School, and she also holds a BA in computer science and environmental studies from Yale University. Her research focuses on indoor exposures to complex mixtures of hormone-disrupting chemicals and builds evidence for healthier materials as a strategy to reduce these chemical exposures in building. So today we have another interesting topic about this silicone wristband. So I'm very happy to have Dr. Young here. So please join me welcoming Dr. Young.
Great. Thank you so much, Dr. Park, and to the M-LEEaD Center for inviting me to talk with you today. It's really an honor, and I'm glad to be able to finally meet some of you. So I'd like to talk today about the results of a recent study that was published just a couple months ago about the hormonal activities of complex chemical mixtures in silicone wristband samples. So first, to put this into some context, in the last decade alone, about 70,000 different chemicals were registered around the world. And a fair amount of those aren't even publicly identifiable due to confidential business information. So we're being exposed to really complex mixtures of chemicals, including many ones that are unknown. Even among the chemicals that we do know about, some of them have hormone-disrupting health effects or endocrine-disrupting effects, meaning that because they look similar enough to natural hormones, they can have consequences for fertility, pregnancy outcomes, development, thyroid function, diabetes, obesity, and certain types of cancer. Now, we're concerned about these because, as many of you probably already know, they're used in all sorts of building materials, consumer products, and personal care products. Now, one reason I think it's been so difficult to effectively reduce our exposure to hormone-disrupting chemicals is this phenomenon of chemical whack-a-mole, where we may phase out one chemical that we learn is harmful to health, only for other similar chemicals with similar health concerns to just be swapped in as substitutes. So we've seen this problem happen time and time again with all sorts of different flame retardant groups, with plasticizers like phthalates and BPA, even with PFAS, stain repellent, and water repellent chemicals. So it's just this endless goose chase of scientists trying to figure out what chemicals are even being used next. And so this is a barrier in our field right now that with traditional targeted methods that test only one or a handful of chemicals at a time, they cannot keep up with the rate that new chemicals enter the market. And so this becomes motivation and challenge to help advance methods that can evaluate our exposures to chemicals as the complex mixtures they are in the real world, not only as individual chemicals. And so our overarching research question for this study was, how hormonally toxic are the complex chemical mixtures that we're exposed to inside buildings? So specifically, our aims were to evaluate the hormone-disrupting potential of complex mixtures of both known chemicals and unknown chemicals that humans are personally exposed to in the real world. So I'll walk through some of the key methods that I've highlighted here. So we started with looking at personal exposures in office buildings. And we use silicone wristbands as these novel passive samplers of exposures where they absorb chemicals from the dust and air and products you're interacting with while you're wearing the wristband. And they can be really useful because they are simple, they're non-invasive, in our case, it was helpful that they don't have to be frozen immediately, so they can be shipped to and from countries around the world. What we loved is that they pinpointed external exposures. So not counting what you're eating or drinking, and in a way control where, when, and how long you're sampling someone's exposures. For our study, we had office workers wear silicone wristbands only during work hours. So they put on the wristband when they got to work, wore it for that work shift, and then stored it in foil and bags overnight. They did this for 4 consecutive work days. And this was a way for us to pinpoint that occupational exposure. So we recruited these 243 people from 36 unique office buildings in cities. So there are about 7 participants per building. And these were located throughout the US, UK, India, and China. And that was from the Global Buildings Study. That was the parent cohort, which is led by Joe Allen. And then our collaborators at Duke, Heather Stapleton and Nick Hergert, analyzed extracts of the wristbands for concentrations of 100 different targeted chemicals, including all sorts of flame retardants, plasticizers, pesticides, PCBs, and PAHs. And then they also conducted suspect screening of the samples to try to tentatively identify other chemical features that are showing up in the samples. And then finally, we cut other pieces of the wristbands and sent them to a lab in the Netherlands, where they conducted hormone receptor cell assays of the extracts of the wristband samples. So basically, they use a human cell line and they put a firefly gene into it such that it lights up in proportion to how much hormonal activity is happening in that cell assay. So I thought this was a cool method that they use a firefly gene, and in that way can measure how much that chemical mixture from the wristband is interfering with estrogen receptor, androgen receptor, and thyroid hormone receptor. So we chose to do these cell assays as a way to rapidly and inexpensively quantify an immediate health indicator of exposures, where you're really linking the chemicals and approximate hormone health indicator that's really close in time. And then compared to traditional targeted analyses, these assays can reflect impacts from theoretically all the chemicals in the sample, not just the ones we know about, or even the ones we can measure in the lab. And then finally, they capture any combined mixture effects. For example, if one chemical has a worse effect when in the presence of another chemical. So for some brief background on how this works, chemicals can be agonists or antagonists. That's one way in which they can disrupt hormones. So for agonist chemicals, basically a natural hormone like estrogen will bind and activate its natural hormone receptor. When a chemical is an agonist, that means it can look similar enough to the natural hormone that it can also bind and activate that hormone receptor. So that means overall there's hormones and chemicals going around activating these receptors, and that can lead to too much activity in the body. When a chemical is an antagonist, that means it can look similar enough to the hormone that it binds to its receptor, but it doesn't activate it. So it basically serves to block it off from being used by the natural hormone. And that can lead to too little activity in the body. So both of these mechanisms can be harmful, especially because these receptors are regulating important genes related to fertility and reproductive health, the menstrual cycle, and sperm production. And in the case of thyroid hormone receptor, growth, metabolism, development, and even brain function. So this was novel in that traditionally cell assays have been applied to one chemical at a time to test its toxicity, or in the case of mixtures, pre-measuring a defined chemical mixture in the lab. There have also been some studies on environmental mixtures like dust in buildings, which we've actually done before. But we're trying to expand that and take it one step further to look at what people's actual exposures are in the environment. And in that way, getting closer, a little bit closer towards the effects for humans. Okay, so I'll first share some results from a previous paper we published a couple years ago that was just on the 100 targeted or known chemicals. So what we found here was that overall there were major differences in exposure by country. So this is 6 different chemical groups here with country on the x-axis, and each color is a different chemical within that class. So without getting too into the weeds right now, if you just look big picture, you can tell that there are pretty noticeable differences by country in the exposures. So I'm just going to walk through a few examples of some of the interesting things we found. So for the PCBs, which have historically been used in building materials like joint sealants and caulking, we found that US participants were definitely being exposed to legacy PCBs that were banned here 40 years ago. So these building materials are sticking around, and that's likely because today half of current office buildings in the US were built before PCBs were even banned. So even this rare success story where these chemicals were banned, the problem doesn't immediately go away. And we see that in our statistical models that newer buildings have significantly lower legacy PCB exposures. Now, what was also concerning is that we found widespread exposure across all the countries to PCB 11, which actually has a loophole in the ban of PCBs because it's an unintentional byproduct in paint and other pigmented materials. So that means it's actually a modern source of PCB that continues to this day. In terms of the flame retardants or plasticizers that can be used in furniture, carpet, electronics, insulation, and other materials, we found, unsurprisingly, that workers in the US and UK tended to have higher exposures to flame retardants, which is likely because of the furniture fire codes that were historically mandated decades ago. Just like the PCB story, we also see that office workers were being exposed to some of these legacy flame retardants like PVDEs that had been phased out over a decade ago. And then there's a later type of PVDE called DecaBDE that actually was phased out later or not at all in some of these countries. And so we see wider exposures to that one. And then finally, even when some PVDEs were phased out, we're seeing that a lot of their newer replacements and substitute chemicals are exposing office workers in these buildings. And last example, I'll just end with some pesticide takeaways. So pesticides can be applied or sprayed directly in buildings. They can also be tracked in from outdoors, or they can be impregnated into building materials. And what we found is that office workers in India were exposed to much higher levels of many different pesticides, such as chlorpyrifos and even DDT. We think this is because they have fewer restrictions of pesticides and more challenges with controlling malaria. So it's actually the only country of these 4 that is still using DDT for that purpose. And then we see that there's a building role here because buildings that reported to have applied pesticides in the last year did have significantly higher worker exposures to certain pesticides. Okay, so those were just some really quick examples. And the overall key takeaways were that office workers were being exposed to legacy chemicals, even if they had been phased out or banned decades ago. In some countries, to chemicals that had not yet been banned, like in other countries. And then finally, to substitute chemicals that are often used to replace the legacy chemicals and have similar health concerns in this phenomenon of chemical whack-a-mole. Okay, so those were about the 100 known chemicals. But in our recent study, we looked more at potentially unknown chemicals. And what we observed were about 1,000 different chemical signatures in the wristbands. About 580 of those were detected in at least half of samples and excluding any duplicates. So if you look just at on average, one individual sample had about 800 different chemical features. And I'll remind you that these wristbands were worn just for 4 work shifts, about 32 hours. So this is, these are complex mixtures that they're being exposed to. Among the features we could tentatively identify sources of, the most common use was reported to be fragrance chemicals. Okay, so that is a lot of data looking at all of these suspect chemical features. So we wanted to tie that into potential hormone interference and prioritize and identify which unknown chemical mixtures are important for our hormone health. What we found in the cell assays was that every single wristband sample was hormonally bioactive. So that was all 243 samples. And I'll note we did do a lot of field blanks that were unworn wristbands, and they were mostly all below the limit of detection. So it's not the base material that's driving these effects, but the chemicals that are collected onto the material. And how we can interpret this is that in human cells, the chemical mixtures that we are exposed to in office buildings can mimic or block human hormones. And in fact, we often saw a dose-response relationship because in the assay, they run 5 different concentrations of the wristbands. So for thyroid hormone receptor antagonism, we saw that 99% blocked thyroid hormone. 96% of samples blocked testosterone at androgen receptor, and about 58% mimicked estrogen at its receptor. What I thought was really interesting is this potential gender disparity, because we found that samples from office workers who reported to be female had exposures that were more estrogenic, more anti-androgenic, and more complex, meaning they had significantly higher numbers of different chemicals in their wristband samples. We think a large reason that helps explain this is that the female identifying office workers also reported to be heavier users of certain cosmetics like perfume and makeup. And we know that cosmetics often contain hormone-disrupting chemicals. However, I do want to note that one limitation of the wristbands is that they could be collecting endogenous molecules from the skin onto the wristbands, and maybe there are some differences by sex there. What we did do to try to control for some of this is we adjusted for squalene, which is in sebum, as an indicator of the amount of skin oil that's being produced and collected onto the wristband. So in all our models, adjusted and unadjusted, these gender disparities were really stark. In terms of the country effects, the workers in the UK tended to be exposed to less hormonally active mixtures. So we can't pinpoint an exact story for why this is the case, but I think it could be due to them having stricter restrictions of certain phthalates and pesticides compared to the other countries at the time. And then for the chemicals that are influencing these hormonal effects, we saw that over 130 different chemical signatures were significantly correlated with each hormonal activity outcome. So this is a volcano plot. You can see these red dots are chemical features that were at least moderately correlated on the x-axis and statistically significant after multiple testing adjustment on the y-axis. So there are a lot of chemicals related to these hormonal activity outcomes. Because we had hundreds of chemicals, many of them could be collinear, we couldn't just run simple regression models. So we chose to use BKMR models as a novel mixture method. And these are nice in that they let you calculate a cumulative mixture effect of all the chemicals on that hormonal outcome. Then they let you identify single effects of individual mixture components on that overall mixture effect. And then they allow flexibility in the relationships, such as them being nonlinear or non-additive. And then finally, you can look at potential interactions between chemicals, although we didn't see any interactions in our study. So the results of our BKMR models showed that the higher the levels of all the chemicals in the samples, the higher the amount of hormonal activity. So this was true for separate models we ran for common targeted chemicals and the common suspect chemical features. So you can see in this graph on the x-axis are all the measured chemical concentrations simultaneously increasing in quantiles. So as all the measured chemicals in the mixture are increasing, we see really strong associations with increasing amounts of hormonal activity. So these are really strong effects that you can see in BKMR models. And then from the BKMR models, we could also identify numerous known and suspect chemicals that were important drivers of those overall mixture effects. So this included several known chemicals such as triphenyl phosphate, a couple phthalates, pesticide, and PCB, as well as other chemical features that we could assign tentative identities to, and then other features that we couldn't identify at all. So among the chemicals we did know source information for, the significant ones included plasticizers, fragrance, sunscreen, and pesticide ingredients. And this is just an example graph of some targeted chemicals and estrogen receptor activation. So you can see how you can get these single effects for one chemical at a time while you're holding all the other chemicals constant. And overall, what these models are showing is that traditional targeted analyses alone are likely missing many hormonally active chemicals. I also want to mention this interesting point about the shared building exposures, because as I mentioned before, about 7 participants were in each office building. So we were able to see that the vast majority of variability in the hormonal activities was due to differences between individuals within the same office building, not to differences between buildings. So what this points to is that exposures can vary a lot within a building. This could be due to different microenvironments. For example, rooms within the building can have different furniture, functions, occupancy, dust loading, cleaning, all sorts of things like that. And as people are wearing their wristband, they may be walking around, kicking up dust and creating this sort of activity cloud around them, this pigpen effect. So that can affect their individual exposure too. And then finally, we saw that personal care products and the chemicals in them are important. So that would be a key factor too. And I thought this was especially interesting that so many cosmetic chemicals were flagged in our study, because it could be that people are putting on cosmetics during office work hours, but it may be the case that, if you put on cosmetics in the morning before work, they could linger on your skin and on your clothes, and thus extend your exposures and even carry them into the building with you. So overall, we found that their exposures were highly complex chemical mixtures, included many usually unknown or usually unmeasured chemicals, showed strong interference with hormone receptor function, demonstrated potential gender disparities, and were influenced by not only building environments, but also personal care products. And I think it also highlights that silicone wristbands are a novel, useful way to pinpoint external exposures in workplaces or other environments around the world. Okay, so I want to end on a more positive and optimistic note and talk a little bit about our healthier materials work. So the idea of healthier materials is to eliminate entire chemical groups that we know are harmful to health. So instead of removing one chemical at a time, just say none of the hundreds or thousands of chemicals in this group can be used. So we had this unique opportunity to study a university that had renovated many buildings with healthier materials free of all types of flame retardants and all types of PFAS forever chemicals, which are used as stain repellents and water repellents. So they had often worked with manufacturers to get furniture and carpet offered as healthier materials for the very first time, because they started this back in 2016. So we were able to collect dust samples in buildings with healthier materials and buildings renovated with conventional materials, and then compare the two groups. What we found is that there were much lower levels of chemicals in the dust from healthier buildings. So there are about 45% lower total levels of organophosphate ester flame retardants, about 65% lower levels of PBDE flame retardants, and about 78% lower levels of PFAS, the forever chemicals. So this was showing that the intervention was working, and that when you act to remove these chemicals from healthier materials, you can actually see a difference. Now, you may be wondering, well, if you can only measure a few dozen types of PFAS, how can you be sure they're really reducing the whole chemical class? So in a more recent paper that was published a few months ago, we went back to the dust samples and measured levels of extractable organic fluorine, because PFAS chemicals are highly fluorinated organic chemicals. So this was a way to get at an indicator of the total content of PFAS, including all types of known PFAS and unknown PFAS, including ones we don't even have names for. And so what we found is that the levels of organic fluorine were also about half in the healthier buildings compared to the conventional buildings. So this was reassuring for the intervention that it was focused on chemical class elimination. Although there, of course, is more work to do to expand these materials to other categories like electronics and building insulation, because they just focus on furniture and carpet. And the way they did this is sort of to focus on the low-hanging fruit first, because we know we can remove flame retardants and PFAS from these products because they're just additives. You don't have to replace them with something else like you might have to for plasticizers. So this was how they started, and they made sure it was the same cost and it didn't affect, project timing. And they have reported that no effects on the performance of the building materials. So this is showing that healthy materials free of entire toxic chemical classes can reduce exposure. So the next step is to make sure that these healthier materials become the default option, not just the exception or something you have to ask to be customized that way. So one thing Harvard is working on for a market solution for this is that they've partnered with other large buying organizations, so like Harvard and Google and other groups, to combine their purchasing power and leverage those millions to billions of dollars to send the market signal to manufacturers that they will only purchase healthy materials. And then it hopefully sends the signal that these should become the norm and the default. So that's what the Harvard Healthier Material Building Academy has been working on, led by Heather Henriksen, the director of the Office for Sustainability. So ending on that more optimistic note, now I just wanted to thank all of our collaborators on this work, especially my advisor, Dr. Joseph Allen, who's director of the Healthy Buildings Program, as well as our collaborators at Duke, UMass Amherst, other Harvard schools, and many other institutions, as well as all of our funding sources. And I think I ended a little early. I must have talked fast, so there's probably some time for discussion. And thank you for listening, and again to the Center for inviting me for this opportunity.
Great, thank you very much. Yeah, we have plenty of time, so and then we already have a few questions. So please, if you have any question, using the Q&A, and then enter your question, and then we will go over your question. Okay, so yeah, this is excellent presentation and very interesting, the results and also your insight. And so we have a few, and then I think you can also look at open the Q&A, and then you can read the question. But for those who will be watching this later, and so we will post this, the the video to our YouTube channel. So I will read the question and then you can answer that. Okay. All right. We have a question from the Jason Golak, and actually we have 3 questions. So first question is, could you explain how the wristbands capture the chemicals? Is it diffusion or surface captures? Are there known interference with this method?
Yeah, thank you, Jason, for your questions. So they can capture chemicals that are semi-volatile chemicals that are both gas phase or particle bound. So one difference in Heather's methods that I'm not sure others use is that she doesn't wash off the wristbands before they're analyzed. So in that way, she makes sure to capture some of the particles on the wristbands, since chemicals could be bound to those particles. So it's not just what's on the surface, but the chemicals actually get into the wristband itself.
Good. Okay. So maybe the follow-up question to this one. So does that mean that, when you ask your participant wear this wristband and then they don't need to change any behavior. So their everyday life and then, shower, washing or physical activity, swimming. So those activity doesn't impact the capturing this chemical using this wristband. Can you
Yes, we
comment on that?
actually have people wear it normally, even during showering and hand washing, because, well, first we might want to capture that exposure to shampoo or soaps, and also because, some of the chemicals are getting into the material. We make sure people know that they can still.
Okay.
That.
All right. And then the next question is, do you believe the graphs provide? And maybe I think this graph is when you show the PCB and PBD and OP and then by different country, so provide sufficient evidence that banning these chemicals is an effective means at reducing exposures below toxic limits, or do you believe that speak at evidence more work needs to be done?
Yeah, that's a great question. I think it's important to ban chemicals we know are harmful to health, but I think we have to make sure to think about how do we remediate it. Once they're banned, what do we do with all these building materials, especially with the aging building stock? Because you have to think about the end effect too. If you're just putting them into landfills, are you donating the building materials to other organizations, and then they're exposing their populations. So I think we have to think holistically about how to make sure the bans actually reduce exposures in the near future.
Great. And the next question is, did you collect information on the cosmetics brands, who were them, etc.? And I think this is related to because you found that male-female difference, and then the women who use more cosmetic and then they have a higher this exposure level, right?
Yeah, so we did. We had a survey every day. They wore the wristband. We asked them all the different cosmetic products they used, and so so we did know that was likely a reason because the female office workers were statistically more likely to be heavier cosmetic users. And we saw that in our previous paper too, where they had higher levels of the chemicals like diethyl phthalate, which we know is often used in cosmetics. But we didn't collect brand information.
Okay. So not brand, but like a specific types of cosmetic relate to the high concentration you found from the silicone band, right?
Right, exactly. Okay. Like perfume, nail polish, makeup.
Okay. Maybe the follow-up question to this question. So then I know. So these days, the PFAS from cosmetic is one serious problem for this PFAS exposure, and also phthalates and other, and maybe the paraben for the preservative used to to maintain the personal care product. So then, do you see that these PFAS and phthalates and those paraben highly detected compared to like a PCB and other like the the flame retardant for those who use more cosmetics?
Yeah, so phthalates were the most abundant chemical picked up on our wristbands, and we did see higher exposures to a phthalate depending on what products you're using. We didn't actually look at PFAS or parabens. I don't think the PFAS were collected well with this sampling method in our study. We actually, Heather and Nick did measure them in the US samples, but we didn't see a lot of detections. So I think that the PFAS and cosmetics problem should be looked at in another way.
Okay.
For that kind of population.
All right. Okay. And then another question, for future research, can you foresee a study using silicone wristband in buildings with healthy building material vs other buildings? Perhaps that could uncover how much cosmetics are influencing results?
Yeah, that's a great idea. Because we were focused on dust for our evaluation of the healthier materials interventions, but that doesn't quite get at each person's actual exposure to dust and air and the products in the building. So that'd be great to look at. And I know Crystal Pollitt's group at Yale is innovating with her fresh air wristband and being able to convert the concentrations of chemicals on the wristband to actual indoor air concentrations. So that could be something interesting to look at too.
So then maybe this is also a follow-up question. So then changing material, that should impact how much the individual who are working that place exposed to this chemical, but also like the air purifier or just circulating the indoor air also impact this exposure, and then that is captured by wristband.
Yes, so that would be really interesting to see, because I could imagine filtration and fresh air could help with particle bound or even fresh air with gas phase chemicals. So there's a lot more work that
because the changing material in terms of the cost and time, so that will take time. So until we actually change and then that intervention happen, maybe the indoor air circulation, so the filtration, that might be a more like immediate intervention method to reduce this exposure.
Yeah, that's true. It's a good short-term solution along with more frequent HEPA vacuuming, things like that. But I think long-term, to eliminate the hazard at the source, it has to come from the manufacturing of the products. And I think that would help with the people who are working and cleaning the buildings and even more highly exposed, along with the upstream geographical areas where these products are being manufactured.
Okay, so and then the next question is from Dr. Stuart Batterman. So nice chat. The wristbands collect both vapor and some particulate phase. Note that dust levels can be quite low in many office buildings. So vapor phase exposure may dominate, particularly for PCBs and legacy pesticide. It would be useful to separate particulate versus vapor phase material as filters can be used to remove much of the former. Perhaps a removable permeable layer on the band.
Yeah, that's a great point. Thank you, Dr. Batterman. So Crystal Pollitt's fresh air wristband, I believe, mostly only does the vapor phase, whereas these wristbands I talked about do both. And I know Crystal Pollitt and Heather Stapleton's groups are working to compare the two methods, and they'll be able to better see maybe how much is the particulate versus vapor phase. So hopefully there'll be more soon about that.
Good, okay. And then another question from Dr. Stephen Brown. So dermal exposure may not predict systemic exposures. So how have been able to make that connection most reliably?
Yeah, that's a good point. So there have been a lot of studies that have compared silicone wristband sampling to blood or urine sampling, and they found strong correlations between the chemicals on the wristbands and the chemicals inside our bodies for several different types of semi-volatile chemicals. So that's reassuring that the wristbands are really capturing and reflecting what can get into our bodies. But again, with the difference that you don't expect them to be perfectly correlated because we're only capturing external exposures without diet or drinking water. So that's important. I am curious to learn more about like how much the wristband prioritizes dermal exposure over air or dust, because like we saw a lot of cosmetic chemicals were picked up in our wristbands and they were the most important drivers of the hormonal outcomes. So I'd be curious if it's, focusing on dermal exposure a little bit more than the others.
Maybe this is also like a relate to this point. And so the result you show, one result I was very stunned was that, so the within building variability was like a 70, more than 70 and up to 99% variability of the exposure, which means that, the exposure captured in this wristband for those participants. So the working hour exposure is dominant one, and then the rest of the exposure happen when they go back home or other place. So that variation is only small portion contribute to that. So which means that no matter how much individual level we try to reduce our exposure, as long as you work in a place where you have no control to change the material and so on, so then you are not able to avoid your exposure.
Yeah, right? That is hard. Yeah. And so these wristbands were only worn during work hours, not the home environment.
Okay.
I expected that people working in the same building to have more similar exposures than we saw, but that wasn't really the case. And I think it's maybe reflected by our previous study where we did these hormone assays of dust in different rooms of some of the same buildings. Sorry, not the same buildings as this study, but the same rooms within a building. And we found that rooms could vary a lot within a building in terms of their chemicals and hormonal activity. So I think just rooms can be really different in a building, and we carry around our individual exposures with us, like through cosmetics.
So then, do you have any study, your study or others, one that, so comparing the workout, so the wearing wristband 24-hour, and so some exposure from the office, and then other exposure from home or outside of the office, and so then from that study design, and do you see what are more dominant or important contributor of the exposure captured in wristband?
Yeah, that's a really great idea. Most wristband studies have just been, the whole 24/7 sampling. So we didn't have a study to compare to that was just the home environment, but it would be cool to look at that.
Okay, all right. Okay, so and then another question from Dr. Batterman. So using a wristband as an area monitor, passive monitor, could also separate the human contribution from the building contribution. Did you consider this type of control?
Yeah, that's really interesting. So Heather Stapleton has done some work comparing stationary wristbands that are just, placed on a table or whatever with the ones people are actually wearing. And what she's told me is that there's way fewer chemicals on the stationary wristbands. So it seems like it's really important that when you're wearing the wristband, you're walking around, kicking up dust, creating this activity cloud, touching surfaces, touching products. So I think she and I were surprised that, stationary wristbands don't capture as much as you might think.
Okay. All right, so we covered the entire the question, and then we have about 4 minutes. So let me take this opportunity. So then you measure various chemical, and then some chemical are very volatile. And so how good this silicone wristband as a tool to measure this volatile VOC chemical compared to more persistent organic compounds?
Yeah, so it's not as good for the volatile chemicals, which maybe is one of the challenges we ran into with PFAS, is that a lot of them are volatile and we couldn't capture those. I think Crystal Pollitt's Fresh Air Wristband does capture, maybe capture more volatility. So yeah, that could be another method if people are interested in those types of chemicals.
Okay, so then like a length of wearing this wristband can help better capture these volatile compounds?
I'm not sure.
Okay.
I don't think it's a matter of that.
Okay.
Yeah, I think it's better for the semi-volatile.
Okay.
Wow.
Okay, and then the last question. Oh, actually, we have one more question. Okay, from Dr. Betterman again. It is also interesting that in a number of studies, home environment are much dirtier than office-type building, school. However, most folks have the opposite impression. So getting at cleaning practice and materials are important. And maybe this is just the comment.
Yeah, that's a really interesting comment. Thank you. Yeah, I think it's not always expected which environment you would think is worse or not. Yeah, like I can imagine homes have denser furniture sometimes. Offices may have more electronics and wiring, and maybe are cleaned more frequently. So yeah, it is a really interesting difference.
Yeah, but I also think because we had in the past, Dr. The Courtney Kerrigan, who talked about her study of the flame retardant exposure in the childcare facility, and then one of the points she mentioned was that because of this childcare facility, and then they have this fire like a regulation, and that's why all the children's toy, not toy, but some of this spongy, the building, and so those are flame retardant are doped with those the product, and there's no way that this childcare facility avoid that, so not using the product. So that's why because the facility has to abide by this regulation, and then they have no choice to choose the different types of material, and then that can be an important source of exposure. So this is very difficult but important point that how we can better this work space in terms of this exposure and other one.
Yeah, it's hard when certain regulations may prevent you from reducing chemicals. I can say that Harvard has done some work on their daycares, and they've been working with manufacturers, and they've had carpet and certain play structures and kids' nap mats have flame retardants and PFAS taken out. So it's good to know that those options are available now for some of those types of products in daycares.
That's good. Okay, so yeah, now 12:50, we are good time. So thank you for your presentation, and then spending this time for addressing all the questions. We have a lot of interesting and challenging questions, and so and also thank you for everyone. Thank you everyone for attending this seminar. So this is our last seminar of this academic year. So I'm very glad that we reached to the end of the semester. So we will see you again in the fall. So thank you, Anna, for your this interesting topic. And
thank you so much for inviting me and for all your great questions from the audience.
Okay. So thank you, everyone. Have a wonderful, still not cold, but wonderful summer, and see you in the fall.